Welcome you to Ciliate signaling systems. We have signal transduction, protein secretion, and trafficking. So if we're ready to begin, I would like to introduce our first speaker, Dr. Ron Pearlman. And he will be speaking to us today, unless he changed the title of his talk, Signaling and Cell Cycle Studies in Tetrahymena.
Okay, there we go. Okay, well, thank you very much. It's a pleasure to be here in this session and to have the opportunity to talk to people. As many of you know, I'm a little bit out of context in this session. Much of our work is on various aspects of chromatin and chromatin remodeling, but we've been doing some work in this system and using some of the same techniques that we use in a number of other things that we do. So want to talk to you about some of these things today. I may be about to embarrass myself. 3 of my students this morning gave terrific talks in the chromatin session, and I may not be able to keep up with them, but I guess that's what we're all about, is hoping that our trainees do better than we do, and I think that's going to happen today. But anyway, I'll try and present this story to you as well as I can. So I won't do any introduction to any great extent here. I can't see at the bottom, but the shared orthologs with humans and the large kinome are the part of the general Tetrahymena introduction that is all that I need to tell you today. So what I want to focus on today, the other aspects that most people in this audience are aware of is nuclear dimorphism, although it's not something I'm going to focus on today. So what I do want to focus on today is protein phosphorylation signaling, particularly phosphotyrosine signaling in multi- and unicellular organisms. And I want to give a little bit of a historical introduction of why we became interested in this and what a little bit of the history is, and then talk about some of the approaches that we've done, some bioinformatics and some experimental work. So phosphotyrosine signaling in response to growth factors is certainly widely believed to be a key signal in vertebrate and invertebrate cells to determine their fate. And receptor and cytoplasmic protein tyrosine kinases are involved in the control of a wide range of cellular processes, such as those mentioned, and I'll mention these again before. But the general view has been that tyrosine phosphorylation, up until, 5 to 10 years ago roughly, that in unicells, there wasn't signaling through tyrosine phosphorylation, and it's really only fairly recently that that's come to fore, and that's what I want to talk a little bit about today. So tyrosine kinases are ubiquitous in signal transduction pathways of multicellular eukaryotic organisms, but as I've just said, it was initially speculated that tyrosine kinases would not be present in unicells. It therefore came as somewhat as a surprise to find tyrosine kinases, true tyrosine kinases, in the unicellular protist Monosiga brevicollis, choanoflagellate, and putative tyrosine kinase sequences have now been found in a number of unicells. So there's quite a literature now. There's a lot of database mining, phylogeny, and things like that. But Monosiga certainly has a lot of tyrosine kinases, true tyrosine kinases and tyrosine signaling, and some of the references are there. So the question is there any phosphotyrosine signaling in other unicells, such as Tetrahymena? And one of the reasons this is interesting is there's a lot of older physiological literature that I won't go into that suggests that ciliates, and particularly Tetrahymena, respond physiologically to stimulation by mammalian growth factor that induce cell survival, activate proliferation, DNA synthesis, chemotaxis, things like that. So that's never been really demonstrated, or certainly in the older literature, it hadn't been demonstrated that that was due to tyrosine, phosphotyrosine signaling, but it certainly raised that question, and people have talked about Tetrahymena as a swimming receptor, really responding to many things. So that raises the question. So the first approach that we took a number of years ago was to do two things: to do Western blot analysis, which I won't talk about, and to do indirect immunofluorescence. What we did, I'm only going to show some indirect immunofluorescence and not a lot. So we used commercial monoclonal antibodies to phosphotyrosine, and we've used the four standard commercially available antibodies, and we've used them in various combinations. They all work. They work together. And we've done appropriate controls. So a question that can be asked is, do these antibodies, which have been raised against phosphotyrosine in mammalian cells, do they recognize phosphotyrosine in Tetrahymena? The controls we've done suggest this is clearly, that they clearly do. So as you can see here, there we go, that 10 minutes after pairing cells of two different mating types, we do see a signal, and that signal is a little bit reduced after further time, but there's clearly an indirect immunofluorescent signal, and we've seen the same signal on Western blots, as I've said, but I'm not going to show any of the Western blot data. The other thing that we've done is there's an inhibitor of protein tyrosine phosphorylation called genistein, and we've looked at the effect of genistein on conjugation, and at the top here you can see that the percent of pairing goes down significantly as you increase the genistein concentration, and pretreatment for 1 hour with genistein and then mixing shows clearly that pairing is decreased. So this correlates with the indirect immunofluorescence that I showed, and this is a genistein experiment, and you can see that on changing increasing the genistein concentration, we get no signal, whereas as I showed you before, 10 minutes after mixing, we get a strong signal by indirect immunofluorescence with anti-phosphotyrosine antibodies. So these were the first things that we did as preliminary things to begin to look into this thing. Their reciprocal induction, which at this point I won't go into, but just again to reiterate that the strongest signals we see are early in conjugation, which may suggest something about a physiological role for this. We don't know this yet, but I'll talk about it, and just this, which is a kind of a nice slide. So protein tyrosine phosphorylation signaling, the general view, hormones, growth factors, cytokines signaling, SH2 protein domains are usually what's involved, protein tyrosine phosphatases, and as I had mentioned before, a number of cellular processes which respond to signaling in organisms where it's been clearly demonstrated. So a few things about some unicells in fungi. In Saccharomyces cerevisiae, there are less than 5 protein tyrosine phosphatases, essentially no SH2 domains except for SPT6, which has an SH2 domain quite conserved, and no things that are annotated as true tyrosine kinases. In the slime mold, similar story, except there are SH2 domains, but nothing annotated as true tyrosine kinases in monocyga. And the reason monocyga is interesting is that it's the purported closest ancestor to metazoans. It's at the border between unicells and metazoans. So emergence of protein tyrosine phosphatases here, fungi, slime mold, which I mentioned, and then true tyrosine kinase signaling here is the way one can look at this. So in Tetrahymena, I'm going to try to go through this story now. I've talked a little bit about the old physiological literature with treatment with growth factors. I've shown you some data with increased phosphotyrosine signaling on mating, pairing of cells. I'm going to go through a little bioinformatic analysis to show you how we've started looking at some of these things. The smoking gun, as it were, is the phosphoproteome that was published by Wei Miao and is available on the Tetrahymena Functional Genome Database, TFGD, and there's the URL for that, and a little more bioinformatics I will go through at the end. All this to suggest that there is protein tyrosine phosphorylation in Tetrahymena and that there are phosphatases, tyrosine-specific phosphatases as well. So the first thing we did was started to do some bioinformatics and looked at domains, tyrosine kinase domains, and blasted them through the Tetrahymena genome and asked what came out from that. And I'm just going to concentrate mainly on the first two, which we call STYK, for serine threonine tyrosine kinase. So these are annotated as dual specificity tyrosine or dual specificity kinases. So they're specific for serine threonine and for tyrosine. And the two that we've looked at mainly are these two, what we call 1 and 2. And the reason we're interested in these is, as you can see, for both of these, the E value is lower for tyrosine in the annotation. If you look at the SMART database or other domain databases, the E value is lower and presumably more significant for recognition of tyrosine, but they are annotated as dual specificity, and I'll tell you how we're trying to look to ask if they are true tyrosine kinases. The others, as you can see, the E values are pretty comparable or higher for tyrosine than for others. So initially, we've concentrated on the ones that have a lower E value for tyrosine than for serine threonine. The phosphatases are true tyrosine phosphatases. That's pretty clear. Single domain that is clearly a tyrosine phosphatase with a very significant E value. We've looked at what we call 2 and 3 mostly, but they're pretty clearly dual specificity, are not very significant at all. So we've concentrated on some of these. This just shows a phylogeny, and again, the 1 and the 2, which I showed you before, which we're going to focus on, tree together and away from some of the others. So whatever they're doing, they seem to be quite similar. So the approach was to understand possible roles of phosphotyrosine signaling by looking at putative tyrosine kinases. I'll tell you why CDK3 and RCK, which are cyclin-dependent kinases. The TKL1 and 2 are these dual specificity things that I had mentioned previously, and the 2 phosphatases. So what we've done is the approach that you've heard from my lab before, and you heard a little bit this morning, engineering FZZ, tag Thermophila strains, and doing various forms of functional analysis. So the idea is to do functional analysis to elucidate function. The first thing we did was pretty straightforward, comparing protein expression data on developmental westerns with the available microarray data, just to look at expression, determine cellular localization pattern, and identifying protein-protein interactions. So the tagging is pretty standard with the FZZ vector from Kathy Collins's lab that many here have used, and it's a standard tagging and transformation into Tetrahymena. And I'll just quickly show you a little bit of developmental expression, so 4 to 6 hours, and a little bit of expression at 8 to 10 hours. This is for CDK3, which in the literature, the Tetrahymena literature, is also called CDC2. It's a cyclin-dependent kinase. And at the bottom, you see the microarray data, so they're consistent. RCK, and I'll tell you why we're interested in CDK and RCK shortly, the same kind of analysis. The protein tyrosine phosphatase is the same thing. This is seen in starved and veg cells as well, and you can see the kind of pattern of expression, and this is PTP3, which is similar. Some indirect immunofluorescence to show localization, and I'll summarize this in a minute. I don't want to go through all of this. So the cellular localization for CDK3, exclusive expression from 4 to 10 hours, localizes exclusively to the mic by indirect immunofluorescence. So possible functional roles in sexual reproduction and in some development. So the putative phosphatases, they localize, well, they're not localized, but you can see cytoplasmic, which is consistent with the indirect immunofluorescence that I showed before. Now, even though these kinases or the dual specificity kinases are not clearly tyrosine kinases, the fact that there really are tyrosine phosphatases suggests that if there are things that remove phosphate from tyrosines, something's got to put them there. So the fact that the phosphatases are there is indirect, but certainly strongly suggestive of tyrosine phosphorylation. So the delocalization pattern is consistent with the observations that I showed you at the beginning, the highest expression during starvation and early conjugation, so possible roles in some of these early developmental processes. So the standard workflow again for interactome analysis, whole cell extracts, looking at our protein of interest, looking at proteins that interact, binding because of the FZZ to an agarose column, washes, elution, and then doing mass spectrometry to identify interacting things. Let me do here first summary of interacting proteins. So we're presently analyzing the CDK3, the cyclin-dependent kinase 3, and I have nothing to say at the moment, although there are some potentially interesting interactors. If you use protein tyrosine phosphatase 2 as the bait, you pull down protein tyrosine phosphatase 3, and if you use 3 as the bait, you pull down 2, so that's quite a robust reciprocal interaction, suggesting that these two may act in concert in such way, in some way, and 2 also interacts with a putative tyrosine with a putative protein kinase, and proteins involved in energy metabolism. So these might, when we analyze them further, suggest further functional roles. So let me go back here and just finish off with another bioinformatic analysis that a master's student in my lab did for her thesis a number of years ago. And there was a paper published, and the reference is on top, by Nett et al. In Trypanosoma brucei, where they looked at tyrosine phosphorylation and phosphotyrosine residues by mass spectrometry in a number of proteins and peptides in trypanosome. Before I can say this, one of the caveats and things that people ask, I had mentioned this before, about whether these monoclonal antibodies are really recognizing phosphotyrosine residues in Tetrahymena. Well, the smoking gun is Wei Miao's phosphoproteome analysis, clearly, where he's demonstrated tyrosine phosphorylation as well as serine threonine, and that database is available. So that's the smoking gun, that there really is tyrosine phosphorylation. One of the things with mass spec, as any of you who have done this are aware, is that you don't get complete coverage. So you don't have all of the phosphorylated proteins coming up, and that's important in some things that I'll mention here. But if you pull out the tyrosine phosphorylations, as we've done, there's 96 proteins in that database that are tyrosine phosphorylated. You can pull these out yourself. I've made a separate database because we're interested in protein tyrosine phosphorylation. If anybody wants them, I'll send you these 96, but you can pull them out yourself from Wei's database. Okay, so back to this. So we've looked at some of the orthologs in trypanosome and asked what happens in Tetrahymena. So the top is a T. brucei, which shows which there is direct evidence in trypanosome for a phosphorylation at this tyrosine here. So this is the ortholog in Tetrahymena. You can see there's a fairly highly conserved region here, and this is the tyrosine in that region. There is no direct evidence that this is phosphorylated in Wei's database because you don't get complete coverage, but it's certainly suggestive that it's very possibly phosphorylated, and we are looking at this by mass spec ourselves. So at the bottom is another protein from trypanosome, which in a very highly conserved region, in fact an identical region, has a phosphorylated tyrosine here, and the identical tyrosine is in fact phosphorylated in Wei's database in Tetrahymena. So here is this direct correlation of that there is a tyrosine phosphorylation consistent with this. And we've looked at many other of the trypanosome proteins, peptides, but this is all I'll present right here. So we've done that. So in progress, what we're looking at, we're replicating some of the affinity purification mass spec experiments to get a better feel of some of the interacting protein. Reciprocal tagging of interacting proteins, I've told you about the two phosphatases that pretty clearly interact. What we're doing to try to address the question of whether these dual specificity tyrosine or dual specificity kinases really use tyrosine as a substrate is by doing in vitro biochemical analysis of the putative kinases and phosphatase and showing this biochemically, also using inhibitor assays in vitro as well as the Genestein assay that we've done in vivo, and further functional analysis using gene knockdown, knockouts, and we do have a knockout of CDK3, which in the Tetrahymena literature is CDC2, so that's being analyzed. We have a knockout. So the last thing that I will show is at the bottom here, and this is another somewhat indirect evidence for functionality. So in the literature, in Schizosaccharomyces pombe, there is, and again, I can't get to it because of the clock down there, but the tyrosine at residue 15 at the bottom, which is the Saccharomyces cerevisiae ortholog, I guess, to the CDK3, P34, CDC2 is, well, it's phosphorylated and it's activated by a phosphatase that removes that phosphorylation at tyrosine 25. You can see, again, quite decent similarity between the T. thermophila, CDK3. It's tyrosine 25 rather than tyrosine 15. It is not phosphorylated in Ways database, but that could be, we're looking at that, but that could be just because the coverage isn't complete. But this again is suggestive that mechanism of action may be similar, requiring dephosphorylation at that tyrosine for activation of the CDK, the cyclin-dependent kinase. So I think with 21 seconds left to go, I'm pretty much done. So what I've tried to show you is the approaches that we're using to, I think, to say from Ways database, the smoking gun is there, that there is tyrosine phosphorylation in Tetrahymena, and we're trying to approach this from a number of different ways. We're not looking at specific pathways at the moment, but using this kind of combination of bioinformatic and biochemical data to try and begin to get at ways of asking functional questions about some of the tyrosine phosphorylation that occurs in this unicell. So thank you very much.
We have plenty of time for questions, if anyone has any questions. I know I have a question.
Sure.
I wanted to ask about, in a couple of the sequences that you showed, there were some tyrosines that were a little bit further upstream of the ones that you had marked in yellow. It almost looked as if there was maybe some type of a similar sequence, maybe a recognition sequence. I wasn't sure if you'd looked into those at all, or if those other tyrosines might also be phosphorylated.
They certainly can be. As I say, the mass spec is limited, that you never get complete coverage. So all I've shown here for the trypanosome data, for example, is where they've demonstrated, where Net et al. Have demonstrated that there are tyrosine phosphorylated sites there, and looked for the conserved region in the Tetrahymena ortholog, and asked, in the one case, in the CDK, that it is phosphorylated in Tetrahymena. In the other case, there's no evidence yet that it's phosphorylated in Tetrahymena. We haven't looked at other things. We've only looked at things where there is direct evidence for phosphorylation, and that comes from the data from trypanosome. So if they haven't shown phosphorylation, we haven't looked at this stage.
Okay.
Okay, Ron, I have a question.
Sure.
So kind of two questions. One, so since you see an increase of tyrosine phosphorylation in this early state of mating, are the proteins in the proteome that show this type of phosphorylation, are they actually highly expressed during early mating? And also you said about the proteome, basically you never get complete coverage. What is the confidence that basically those tyrosine phosphorylations are actually real hits?
So two questions. We don't know what the proteins are that are phosphorylated in the early stages of conjugation. We haven't looked at... Oh, this is in Ways database. There are 96. Yes, I can't. We've looked at that. I can't remember offhand. I'll have to go back and look at my data. There are, but I don't remember how many, and I don't remember which ones. But we have some Western blot data from the same time points as the indirect immunofluorescence, but we haven't identified specifically what proteins are because those are from whole cell Western blots. So we're trying to look at that, but we haven't. And what was your second question? Oh, I think you'd have to weigh that, but I think from the methods that he's used to identify phosphorylated serine, threonine, and tyrosine, I think it's very robust that those are phosphorylated if you look at the methods that he's used, but you don't get every phosphorylated site in a database like that. Does that answer that? I hope.
All right, well, let's move on to our next talk. Let's thank Dr. Pearlman.